Today, I want to present some data from Germany, and this involves targeted metabolomics and across different prospective cohorts in Germany. But before I start with the science, I want to tell you a little story. So Annette has already been talking about the early times in metabolomics and cohort studies. And what you can see here is a flyer of the first international scientific symposium on metabolomics in general in Germany. So this symposium was as you can see here, in 2010. And this, by coincidence, happened to be my first conference as a fresh PhD student. And I was really excited to be there. And during dinner time, I had the chance to sit right next to one of these mass spec experts from the United States. And I told him all about our project, that we were planning to do metabolomics in a large cohort study. And you know what he replied to me? He told me, well, you guys must be crazy. And I really will always remember what he said, because now, 11 years later, we are sitting here and talking about this crazy stuff, and so many people are interested in this topic. I find this just really, really fascinating. So, but now I want to move on with the science. So in large cohort studies, what we want to do usually is we want to measure as many metabolites as possible, and then in prospective cohorts, link this to the incidence of, for example, chronic diseases. And now, what is the advantage of metabolomics in cohort studies? Well, what we usually have in cohort studies is this one really valuable baseline sample. And everyone wants to measure all kinds of markers in the sample. So when we use metabolomics, we are very efficient because we only need very little sample volume, and we can measure many markers. So in the case of targeted metabolomics, this is usually 1 to a couple hundred metabolites. And also with targeted metabolomics, we have the identity of the metabolites, so we know what we are measuring. And often we have even quantitative data. So what do I want to talk about today? I decided I want to go a little bit more into detail and also talk about the study designs and the methods that we use. And then I want to present you today results from 2 studies. One is concerned about risk of cardiovascular disease. This was done in 2 prospective cohorts. And the other one is concerned about some network analysis. And we did this in 4 prospective cohorts in Germany. So to start with our first part today, this was done in the EPIC study. This is a European cohort with 500,000 participants. And in Germany, we have 2 study centers in the beautiful cities of Potsdam in the east of Germany and Heidelberg in the west of Germany. And in total, we have their blood samples of more than 50,000 participants. And we followed them over time for the incidence of chronic diseases. So in my case that I want to present today, cardiovascular diseases, that we had a mean follow-up for this analysis of 8 years, but we also have the other chronic diseases like cancers and type 2 diabetes. And now when we want to do metabolomics in these large cohorts, unfortunately, we cannot measure the full cohorts because it is just really expensive. So what we did is we drew some random subcohorts in each cohort, and then we created a so-called case-cohort study. This means we have all the incident cases of the full cohort, and we use the random subcohorts as control group. And the advantage of this approach is that you can use the same control group for many diseases because it was drawn randomly. So, and then you can just measure metabolomics in the subcohort and in the cases, and you can do your analysis. And we use targeted metabolomics. We used the kit of a well-known company. We measured about 160 metabolites, and we used different labs in the 2 cohorts. Well, in terms of statistics, I also want to talk about this because I think this is also an important part. And really, this is also sometimes a problem because the data is just so complex. So in this study, we used a really simple approach, I would say, or a classical epidemiological Cox regression. modified after apprentice because this is a case cohort design. Our exposure were 105 metabolites, and we had 2 outcomes, stroke and myocardial infarctions. We ran 2 separate analyses. And then we had to pick like a model. So we included the classical CVD risk factors, age, sex, smoking, alcohol, and so on. We also ran some additional models where we adjusted also for some classical CVD biomarkers. Well, because you ran so many tests, what you have to do is you have to correct for multiple testing. And then additionally, we had the problem we could not combine the data from the 2 cohorts in one data set. So what we did then was a meta-analysis across the 2 cohorts. And we said we only say a metabolite, we only select the metabolites that are consistently linked with cardiovascular disease risk in both cohorts. We also calculated some measures of risk prediction, and later on we went into some network analysis, and I will talk about the network analysis in the second part. So now let's come to the results. For the association between serum metabolites and risk of stroke, it's said in 2 words, we found no association, unfortunately. So of course we found some associations in one cohort and others in the other, but none of them were consistent. Well, luckily for us, we found some associations for the risk of a myocardial infarction. So actually 10 of the metabolites were consistently associated with higher risk of myocardial infarction in both cohorts and also independent of these classical CVD risk factors. So what you can see here now are our forest plots, and you see the metabolites, and then you see the 2 centers, and then here's the risk estimate and a pooled risk estimate. So the risk was about 40 to 50% higher risk of myocardial infarction per standard deviation increase in these metabolite concentrations. And when you look at the metabolites, you have here some diacyl-phosphatidylcholines, acyl-alkyl phosphatidylcholines, and some sphingomyelins. Well, we went on and were asking whether these associations are explained by some classical CVD biomarkers. So what we now did is we added to our basic risk factor model these classical biomarkers that we had also measured in the cohorts, like HDL, LDL, total cholesterol, triglycerides, AHS, CRP, and also one model where we included all of them together. And we ended up with 3 metabolites that were still consistently associated with risk of myocardial infarction in both cohorts and independent of these biomarkers. When you talk about these associations and you have this longitudinal data, there's always the question of risk prediction. So is there a clinical utility of these metabolites? So we drew some ROC curves, and the area under the ROC curve is the probability that a true case is assigned a higher risk compared to a control. And here you can see now our different models, the basic risk factor model, and then what happens when you add metabolites and biomarkers and all of it. And you can see you get a little increase in the prediction, but I would not say this is so much and this is so much more information. So just to summarize the results from the first part, none of the metabolites, surprisingly, were consistently associated with higher risk of stroke in these 2 cohorts. However, 10 of them, particularly the phosphatidylcholines and the sphingomyelins, were associated with higher risk of myocardial infarction. And 3 of them were also independent of classical CVD biomarkers. Well, we were not really satisfied with these results, and we were thinking, well, now we look just at the single metabolites and we just ran like 100 Cox regressions, but is there sort of a modeling approach to get a picture of all metabolites and their interconnections? So I really like this metaphor of a cherry tree. What we did here in this study is we picked out the really bad cherries, but what you actually want to know is what's wrong with the tree. So, and yes, there is, you can use metabolite network analysis. And I want to go into a bit into detail here. So just metabolite network analysis in a nutshell today. We use Gaussian graphical modeling. And usually when you have metabolomics data, your metabolites are highly correlated. And it is really difficult to sort out the meaningful and true correlations. So what you can do is you can use metabolite A and metabolite B and calculate the partial correlation between the two, adjusted for all the others. And then you sort out only the strong partial correlations that are left behind. And these are some true associations or some direct effects. So, and when you have a network, you call the metabolites nodes, and the connections, so the partial correlations between metabolites, are the edges. And this is a data-driven approach, but when you run these models, you actually find some biological plausible associations. So usually the metabolites that are really neighboring in your network, they are one reaction step apart. So what you can see here is phosphatidylcholine with 32 carbon atoms, and the other one with 34 carbon atoms, and this is one elongation reaction. So we did this in a previous study in an EPIC-Potsdam data, and here you can see the network of the EPIC-Potsdam cohort. And what you can see is that these metabolites cluster really by their chemical classes. So you have the amino acids in one cluster, acylcarnitines. You have these phospholipids based on the chemical classes. Now we went one step further, and we also entered one more information. Then this is the association with some determinants of metabolites. So here we used waist circumference, for example, which was also measured in the EPIC-Potsdam participants. And the color now of the metabolites depicts the association with waist circumference. So all the red metabolites positively associated with waist circumference, all the blue metabolites are inversely associated. And what you can see in the middle of the network, for example, you have here some red cluster of metabolites, and there's one really dark red dot. So actually, when you look, which one is it? Well, this one is diacyl-phosphatidylcholines C38-3, and this one is highly positively associated with waist circumference. And when you remember my results from the first part, this one was also strongly associated with risk of myocardial infarction. And I tell you what, it's also associated with future risk of type 2 diabetes. So I guess here we picked a really bad cherry. Then the question came up, if we do these network analysis, if these are really biological pathways, are these pathways or are these networks similar across different cohorts? Wouldn't this be actually a prerequisite if you want to pool metabolomics data from different cohorts? So we thought of how could we approach this question? And we looked around to our neighbors in Germany and looked for other cohort studies that also had targeted metabolomics data available. And we found two other cohorts, the KORA cohort, which is conducted in Augsburg. And also Annette was talking about results from this study. And there we had data from 3,000 participants. And also the CARLA cohort. This is conducted in Halle, close to Leipzig in the east of Germany. There we had also metabolomic data. And these four cohorts now were all a little bit different. You can see a little bit different age. So CARLA participants were the oldest, also had highest BMI, and were more likely to be men. And now what did we do? Well, we looked what metabolites were measured in these cohorts, and we found an overlap of 100 metabolites that survived the quality check. And then we constructed metabolite networks based on the method I just presented for each cohort. Then we said, okay, for each cohort, the same 100 metabolites are included for the network construction, and we've selected the 100 highest partial correlations between the metabolites as edges. And then we just compared the networks and also did the meta-analysis. So here are the results of this analysis. This is the common metabolite network of the four cohorts. This means we picked out all the metabolites that were the same in the four networks and all the metabolite connections that were the same. And what you can see here, for example, also some biological plausible reactions, just like a desaturation reaction. And when you look at our definition, what we put in the model, well, 66% of the metabolites and 57% of the metabolite connections could be reproduced in all the 4 cohort studies. We went then on and took a closer look at the edges. So the edge is a direct connection between the 2 metabolites in a network. And we said for each cohort, we allow the 100 strongest edges. And here you can see the 4 different cohorts and the edges overlap. What you can see, all 4 of them have a 57% edges overlap. But when you look, for example, at KORA and Potsdam, you have 83 edges overlapping. And KORA, CARLA, and Potsdam, for example, together, they have an edge overlap of 76. Now, we had also some other measures. We looked at the Hamming distance. Well, the Hamming distance tells you how many changes you have to do to convert one network into the other network. And the lower the Hamming distance, the more similar are the networks. So for example, CARLA and Potsdam, you have to do 7 changes to convert the networks. So they are quite similar. On the other hand, on the lower side here, we looked at the correlations between the networks. And also you see, for example, Potsdam and CARLA have a very high correlation of 0.96. However, what you also see, you would expect that Potsdam and Heidelberg are quite similar because they are from the same large cohort, just different study centers. But you see they have the lowest correlation of metabolite networks. And one reason for this maybe, well, CARLA, KORA, and Potsdam were all measured in the same lab. Unfortunately, Heidelberg was a different lab. And you can also see CARLA and Potsdam, they used the same kit. So you can say the networks are most similar when measured in the same lab and also with the same kit. And I think this has also some implications for future studies. So then in the end, this is now my last slide for the results. We drew a meta-analytic network of the 4 cohorts. So how did we do this? For each network, the backbone is a correlation matrix. And what you can do then is to just do a meta-analysis of the correlation coefficients. And then you get some pooled correlation coefficients. And then you can go back and draw a network. And this is now the meta-analytic network. And all the metabolites colored in red here are the ones that were only found in the meta-analytic network compared to the network that was done based on all the similar metabolites and edges. So when you do the meta-analysis, you gain information and you get a total of 89% of metabolites connected in this network compared only to 66% when you only use the ones that were really the same. So to summarize my second part, it was possible to pool metabolomics data from 4 different German cohorts and also to draw a common metabolite network. And the metabolite networks of the 4 different cohorts were most similar when measured in the same lab and also with the same kit. However, I think that meta-analytical approaches are quite promising towards combining this targeted metabolomics data from different prospective cohorts, and particularly when they are measured with different, in different labs and with different kits. So what is now the outlook for future cross-cohort metabolomic studies? Well, it would be great to include more cohorts, also to increase the diversity of the cohorts. So this study was just done in German cohorts, but you could go, for example, for Europe, also for Asian or globally. Also, the age range could be increased. Yeah, usually you have only one time point in these cohorts, and this is always only a snapshot. It would be great to include more time points for each participant, and also eventually to expand this to untargeted metabolomics. But I think this will be really challenging. And yeah, we are addressing all these things right now in an ongoing work. And I'm now a PI of a project where we do untargeted metabolomics at multiple time points in 2 children cohorts. And this project is in collaboration with Augustin Scalbert, who will also talk today. I just had to think about what John said. He said, it is always our parents' fault. So yeah, he's true. And that's why at BIPS in Bremen, we are part of a large European children cohort, the so-called EDIFIX I-family study. And we use data and biosamples of 8 countries in our project. We have great data here, not only from the children, but also from the siblings of the children and the parents. So this is a really great cohort that was set up. Also, we use another children cohort to validate our findings. And this is the Donald study. This is a regional open cohort study supervised by the University of Bonn and Professor Ute Nödlings. And I think we will get really some great data out of this study. So this is my last slide today. What are the challenges? Well, you need some sort of overlap of metabolites to combine data from different cohorts. And this should be like a minimum of overlap, I think. Reproducibility is a really big challenge when you use different labs and different kits in the cohorts, but also you have different assessment methods for all the other variables that you are interested in. But of course, yeah, you want to increase the diversity of cohorts, but this is always also the challenge. So I think it's more or less a trade-off on how we need some high reproducibility, but also we want to increase the diversity. This is the trade-off we have to think of. One point which I find also really important is the data accessibility from different cohorts. So in our project, we were fortunate enough to get the original data from each cohort, but this is usually not the case. And of course, these data sets are getting quite complex. So with this, I want to finish today. I want to thank everyone who was involved in these past and in our current projects. Much of it has been done at the DIFE in Potsdam. Also, our future projects here at BIPS in Bremen, I find great. So if you have any questions or if you want to collaborate with us in the future, you can just write me an email. Thank you.